ATP is really great, but you wouldn't want to get it too late. So your muscles that need ATP fast rely instead on creatine phosphate. So ATP is like an energy storage molecule and it's really great. It can power all sorts of reactions, but you don't want to have a ton of it lying around because you have all these sorts of reactions happening all the time. So your bodies instead store up on things like lipids and carbohydrates, things that they can break down to make ATP when they need it. But if you need that ATP really fast, well then you don't want to wait around for that breakdown. So your muscles use an alternative source that is closer to ATP. So it's only like a single step removed. This creatine can get creatine phosphate, can transfer the phosphate group onto ADP to make ATP much faster, and this allows your muscles to have a source of energy. And your cells also have ways to control the levels of ATP. And so let's talk about all of these different things.
So the other day, I talked about how ATP is kind of like a cellular energy currency. That's kind of like an arcade token in the sense that your body can take in sources of energy from all different kinds. So we're talking lipids, so like fatty things, proteins, sugars, and your body can break down these molecules and take the energy from these molecules and convert them into these molecules of ATP, which then different like molecules in your body can use to help pay for different reactions. And so how ATP does this is the phosphate groups of ATP. So ATP stands for adenosine triphosphate. So you have this adenosine and then these three phosphate groups. These three phosphate groups are negatively charged. When you hold these negatively charged molecules together, well, that's really uncomfy. And so this is going to take energy. This is going to be what we call like a high energy bond because it has high potential energy. It's kind of like clamping a spring. It takes energy to hold that spring clamped. If you let go, well, now you get that energy back that you were using to clamp because of the whole like laws of thermodynamics. There's no way you can't make or destroy energy. You can only transfer it in different forms. So you have energy that you're using to clamp down the spring. You have energy that's held in these bonds, holding these negative charges together. Remember, like charges repel, so these are repelling another. You're holding them together in the chemical energy of this bond. If you then break the bonds, now you get that energy back. So if you were to release the spring and you had something like, say, on the end of the spring, well, now that would fly across the room. But in the case of these molecules, instead, we use the energy to power reactions. So you might think, okay, well, if you have a reaction and you have some reactants, your starting material, and they're going to products, your finishing material. Often the products are going to be a lot happier than the starting materials. And you think, okay, well, that's just going to happen on its own, right? And in fact, if this is the case, if these products are happier, if they're more like energetically stable, we call these reactions spontaneous. But they're not just going to happen all the time because it typically takes some sort of activation barrier that you have to get over. So just like if you're sitting on a really uncomfy chair and there's like a couch across the room, you might be more comfy in that couch, but you have to actually get up and go over to the couch. And so there are molecules called enzymes, which can help make this lower that activation energy in order to make it easier to get to the other side. Often these enzymes use help to do it. They get help in the form of this ATP. Additionally, ATP can also provide energy to make reactions that aren't spontaneous, so reactions where the products aren't at a lower energy. They can make these reactions happen. So enzymes catalyze reactions in both directions. So by catalyze, I mean that they speed up the reaction without being used up in the process. So if you imagine a stick snapper, then it would be kind of like snapping the stick would be the enzyme's job, or that would be like the overall reaction would be to snap the stick. The enzyme might stabilize it in its almost snapped position, so helping make comfy the uncomfy part, this like transition state. But it would also be able to, because it's holding this together, it can then promote their rejoining. And so enzymes will catalyze a reaction in both directions. And importantly, when it comes to ATP, we can use control the levels of ATP in order to help determine which direction a reaction will go. And this is going to come into play when we talk about how we regulate levels of energy and regulate which form of ATP or ADP we are storing. So enzymes, lots of different enzymes can use ATP. That's one of the great things about having this like kind of universal currency. Doesn't matter where that ATP originally came from, all these different enzymes can use it. But the problem is that if you have a ton of different enzymes that are all capable of using this ATP and doing all these different reactions, if you just have a bunch of ATP sitting around, you're going to have a bunch of random reactions going off in your cells all the time, which would not be good. So your bodies have to carefully regulate the levels of ATP, and instead of stocking up on ATP, they stock up on the energy storage forms. So they stock up on those big lipids. They stock up on those long sugars like glycogen, and then they break them down as needed. And we'll talk a little bit later about how your bodies can actually help regulate the levels of ATP and ADP in order to keep a nice balance so that you are making and breaking molecules when you need to do so. But it takes time in order to actually make ATP. Your body has different ways of making ATP depending on the starting material, but often what we're talking about is cellular respiration. Sometimes it begins with glycolysis. It can also begin with different things. But the main payout happens in this process called oxidative phosphorylation, which happens in mitochondria, which are these little membrane-bound compartments inside of your cells. In this process of oxidative phosphorylation, you produce a lot of ATP, and it's this really cool mechanism involving like these tiny little pumps and various things that I have more on in another post. But it's a multi-step process, and you have a bunch of steps before that, depending on your starting material, in order to get there. So long story short, this takes a lot of coordination, a lot of steps. And if you need to run away from some like monster that's running at you, you want something that's faster, which is why in our muscles and some other cells, they don't want to wait for the ATP to be formed in this long pathway. And so instead, they need to have a quicker source. But they still have that problem where they wouldn't want to stock up on ATP because the different enzymes are specialized to use ATP. So remember, it's kind of like an arcade token. So all of your arcade games would have a slot for this ATP arcade token. But they wouldn't have a slot for a, say, phosphocreatine arcade token. So you can store like potential energy in this form that's a lot easier to get to ATP from, but those enzymes still can't use it. They still don't see it as like ATP. And so how our muscles do this in some other cells is by using creatine and phosphocreatine or creatine phosphate. So sometimes you see this written like CRP, sometimes PACR, that you see various abbreviations for phosphocreatine and creatine phosphate. And this comes from phosphorylating creatine. And this phosphorylation is done by this molecule called creatine kinase. So a kinase is a molecule that adds a phosphate group, and kinases typically add this gamma, so this last phosphate group, from a molecule of ATP onto a molecule. And so we've talked in the past about how how kinases can phosphorylate proteins, say. And so this is a form of post-translational modification. So the process of putting together amino acid building blocks to build a protein is called translation. And so if we have modifications that happen to those protein, to the residues from the amino acids, so when you link together amino acids, the leftovers are called residues. And then when you can modify those through post-translational modifications, and these include phosphorylation. And these phosphorylations are carried out by molecules called kinases. But in addition to phosphorylating proteins, there are also kinases that specialize in phosphorylating small molecules, such as creatine kinase, which phosphorylates creatine. We'll talk more about what creatine is in a little bit, but this is creatine, and it's a small molecule, and it gets phosphorylated by creatine kinase to form phosphocreatine. So what did you do here? So, well, now you have ADP left over as well as this phosphocreatine, and it has the potential to go back and make ATP because remember, enzymes catalyze reactions in both directions. They lower the barrier to get from the reactants to the products, but that also means that they're lowering the barrier to get from the products to the reactants. So which the reaction is going to go is going to depend in large part on the concentrations of the products and the concentrations of the reactants. So if you think about how this reaction is going to go, the enzyme is going to help it in both directions. But if there's more of these things to bump into each other, they're more likely to go this way. And if there's more of these things to bump into each other, there's more likely to go this way. So you can imagine an extreme where you have no ATP. If you have no ATP, then you can't go this way at all. And so the reaction would be driven back this way. So this is what happens if you have low ATP levels. You're going to make more of ATP. And this makes sense because, well, if you're running low on ATP, then it makes sense that you would need to make some ATP. And so the concentrations are going to promote the making of more ATP under these conditions. Now let's look at the other extreme. What if ATP levels are really high? So you have a lot of this, and say maybe you have none of this, or you have a little bit of this. Well, now you're going to have the reactions. These guys are going to be bumping into each other more, and they're going to be, when they're bumping into each other more, with the help of this kinase, then they're going to be driven to go in this direction more. You're going to end up with more of this phosphocreatine and this ADP. So in this way, you're using up that extra ATP, so you don't have a lot of extra ATP just hanging around. But you're not going to convert all of the ATP because this reaction is reversible, and you're going to, once you start building this up, then you're going to have more going this way. And the bottom line is that you end up with a nice balance of your ATP form as well as your phosphocreatine form. And this is going to allow you to tightly control the metabolism that's going on. So metabolism is the making and breaking of molecules. So this encompasses anabolism, which is molecule making, as well as catabolism, which is molecule breaking down. And so this can be regulated by the concentrations of ATP in this manner, as well as we'll see how it can be modified, be used in other cellular contexts to monitor the ATP levels can be used in other cellular contexts in order to regulate metabolism as well. But this creatine phosphate is how your muscle cells, as well as some other cells, as we'll get into, but mainly your muscle cells. Yeah, so it also in brain, retina, sperm, but the muscle is the main creatine user, so about like 94% of your body's creatine is located in there, and we'll get more into some stats about it later. So let's talk a little bit more about what creatine is, and then we'll get into those stats. So creatine itself is actually technically an amino acid. So we typically think about amino acids as like these protein ladders or these protein building blocks. And I talked all about these yesterday and in a lot of past posts, so I'm not going to get into them here. But these are the proteinogenic amino acids, so the ones that are actually used that get linked up into together through peptide bonds in order to form proteins. But really the thing that makes them an amino acid is because they have an amino group and they have our carboxylic acid group, which typically is deprotonated in this carboxylate form. So an acid is something that donates a proton. Similarly to how we had our enzyme reactions go back and forth, this can go back and forth even though it's not enzymatically mediated. Basically, you can have protonation and deprotonation that goes from the carboxylic acid form to the carboxylate form. And so it's just kind of like a terminology thing where an amino acid has an amino group and then a carboxyl group, which could be in the carboxylic acid or the carboxylate state, depending on the pH. I'm not going to go into that here, just in terms so you don't get confused about the nomenclature. So this is all about an amino that something takes in order to be an amino acid. And so let's look at the creatine and see if it qualifies. So creatine, well, it's got amino groups and it has a carboxylic acid group. So yes, it is an amino acid, but it's not one of the amino acids that we find in our proteins. But it's made from amino acids that we find in our proteins. In fact, making creatine requires 3 of those like normal amino acids, so 3 of the proteinogenic ones. Glycine and arginine, and then methionine indirectly. So we have glycine, we have arginine, and then we have methionine, which is going to be used to provide this methyl group, but it's not going to be like, it's not part of the core scaffold. And making this is going to happen in 2 main steps. One of these is going to happen in your kidney. The first step happens in your kidney. Then the second step is going to happen in your liver. And then it's going to release this creatine in different cells. The cells that have the receptors for the creatine can take it in. And so this is why your muscles end up getting most of it, because your muscles have those receptors for it, and so can take in this creatine. And then your muscles already have the ATP and stuff. And so then they can, and the creatine kinase, and so they can use this creatine phosphate storage system. So how this reaction actually happens is, so it's helped out by a couple of enzymes. So in the first step, you have this GAT, arginine glycine aminotransferase. And so basically this N group of arginine is called an amidino group. And what happens is that you take glycine, you take arginine, this enzyme goes, voila, it gives the amidino group from the arginine onto the glycine. And this leaves you with ornithine, which can be used in other metabolic things, as well as this guanidinoacetic acid. This can then get exported from the kidney and go to the liver. Now, in the liver, there's a different enzyme. And this enzyme is called GAMT, or guanidinoacetate methyltransferase, which is going to move a methyl onto it. So move the CH3 group onto it from S-adenosylmethionine. Sometimes called Adomet. We'll talk more about this in a second. This is a common molecule that's often used as a carrier for methyl groups. Here, this methyl group is going to get added on to your guanidinoacetic acid, and this is going to give you creatine. This creatine then gets released from your liver, and those cells with the receptors for it can take it in. So you have your arginine and your glycine are going to be the main sources of the scaffold, but then this methyl group is going to come from methionine. So indirectly you use methionine, but methionine can be made to make S-adenosylmethionine. And then this molecule can actually be used to methylate all sorts of different things. So it's not like methionine is like specifically used in this reaction, but rather this product that comes from methionine. So methionine is really, really cool in that you make this, that you can use this group for methylation, because it kind of has this like random this random methyl group just like hanging off of a sulfur, and sulfurs can be more reactive and vulnerable to have this taken off and things like that, and much more on this in my post on methionine. So let's talk a little bit more about creatine. So as I mentioned, that first step is going to happen in your kidneys, and then the second step happens in the liver and then releases it into the bloodstream. And then only the cells that have the receptors for it can take them in. So these cells have like these special transporter proteins in their membranes. So our cells are surrounded by these double membrane, this like phospholipid bilayer. And in order to get through that, this creatine has to go through a special transporter. And only certain cells have these transporters. And so the cells that express these, so all the cells have the instructions for making them, but they don't all make it. Because they don't need them. And so the cells that need them do express this. So they do make this protein, and they have this protein in their membrane, and this protein in their membrane is going to allow them to take in this creatine. The cells that have this include the muscles, so your skeletal and cardiac muscle, so your heart and your muscle, your like the muscles you think about as muscles. Your brain, your retina, so in your eyes, and sperm where applicable. But the muscle is the main user. And so about 94% of our body's creatine is located in there. And we have a lot of creatine, so about 120 grams of total creatine. But you keep needing to send more in because about 2% of that creatine gets actually like non-enzymatically made worthless. So basically enzymes make reactions happen, but it's kind of like you can lead a horse to water, but you can't make it drink kind of thing, in that the enzyme isn't going to it can't make a reaction happen that is like impossible, and it can't, it can only make reactions like more likely to happen, such as, say, holding molecules together in the right orientation to react or that sort of thing. But if those molecules don't want to react, they're not going to react. Similarly, those molecules could react on their own. It just might take a really, 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 really long time because those molecules all floating around randomly would have to find each other, bump into each other in the exact right way and in these like ideal circumstances. And so the enzyme makes it a lot easier to happen, but it doesn't mean that it can't happen. And there are some reactions that can easily happen without the help of enzymes, and there are other reactions that can't easily happen without the help of enzymes. One of the reactions that could happen without the help of enzymes is this kind of like non-enzymatic circulation reaction where you go from creatine to creatinine. You get this kind of like circulation. This product is no longer useful in this for energy storage. And so your kidneys are going to get rid of it, and they're going to filter it into your urine, so you pee it out. So doctors often measure creatinine levels in your urine, such as like over 24 hours, and compare this to the levels in your blood to get an idea about your kidney function. So they don't care so much about the creatinine, but rather your kidney's ability to filter it out. And so by comparing your blood to your pee, your doctors can say, okay, well, are your kidneys having problems filtering out molecules? And they can also use it to measure things like glomerular flow rate, so how fast things are moving through your kidneys, and various things like that. Yeah, so this 24-hour things allows you to compare to check creatine clearance rate, which they can use to estimate the rate of blood flow, so this glomerular filtration rate. This gives an overall idea about the kidney function. A little bit of history. So it was discovered in skeletal muscle in 1832 by French scientist Michel Eugène Chevreul, who named it with inspiration from the Greek word for meat, kreas. And in 1912, Otto Fallon and Riley Glover Dennis found that if you give animals a lot of creatine, it mostly got stored in their muscles. And in 1927, the phosphorylated version was discovered by groups at Cambridge and Harvard. And then the source of the phosphorylation, so that creatine kinase reaction, was discovered in 1934 by the German biochemist Carl Lohmann. So that reaction is sometimes called the Lohmann reaction. We now actually use phosphocreatine and this creatine kinase to our advantage in the lab when we're doing in vitro reconstituted translation or lysate-based reactions as well. So when you're doing like cell-free protein expression, so trying to express proteins outside of a cell, you typically use you need an energy regeneration source. So a common system is this pure system, which is an E. coli-based system made up of reconstituted from purified components. So you have your coli ribosomes, tRNAs, translation factors, and then importantly, you have this energy regeneration system. This energy regeneration system involves creatine kinase and creatine phosphate. And so in the system, it's able to regenerate ATP quickly because in the system, you're working outside of cells. You don't have those mitochondria. You don't, even if you could wait around for the cells to make it or for the system to make energy, well, if you don't have mitochondria, well, then it's a moot point because you have to wait and like forever because you don't have the machinery there to actually do it. And so by adding creatine phosphate, and creatine kinase, you can then get this reaction to give you an alternative source of regenerating that ATP that you're spending in that translation reaction. So pretty cool stuff. As I mentioned, it's only certain cells that actually have the ability to regulate, to store ATP in this manner. But your other cells need to kind of regulate via ATP and regulate their ATP levels as well. And the way that they can regulate their ATP levels involves a lot of feedback between different enzymes in the pathway. So we talked just to go back to when we're looking at the different ways that our cells can make energy. So in like cellular respiration, you have glycolysis where you're breaking down like sugar. The pyruvate oxidation, you have this thing called the citric acid cycle, and this is all making components that feed into this oxidative phosphorylation pathway. Different steps in this can be regulated based on ADP or ATP. So if you think about what you would want to happen, if you have a lot of ATP, you're going to be wanting to use that ATP. You're going to be wanting to do anabolism, so make molecules. If you don't have very much ATP, then you're going to do catabolism. You're going to break down molecules in order to make ATP. So typically when you use ATP, you end up with ADP. So this is kind of like the product of actually using ATP. And so this would be like our reactant and this would be our product, or depending on which direction you're looking at things in. But we have that same thing where we're going to have relative concentrations helping regulate things. And so this regulation can be directly, such as this by altering the concentrations of these various components makes it more or less likely that these reactions can happen. But the regulation can also be happening by ATP or ADP actually binding to and directly like changing the activity of the enzymes that are involved in the energy making processes. So for example, ADP allosterically activates glycolysis in TCA and ATP allosterically inhibits glycolysis in TCA. So by allosterically, what that means is that these molecules are binding someplace and other than like the active site, so other than the site on the enzyme where things actually happen, it's binding somewhere else and kind of like changing the shape of the enzyme so that it makes it more or less reactive. And you can also have things like phosphorylation. If you have a lot of ATP, you'll get more phosphorylation, and that can also allosterically regulate enzymes. And so there are different ways that these enzymes and these processes can get regulated to help regulate the overall usage of energy and the amounts of ATP you're making, the amounts of ATP you're storing. And in this way, you're able to keep good levels of ATP in all of your different cells. So you don't have those like reaction fireworks going off all the time, but you still have ATP where you need it. And in your muscles, you can get ATP more quickly using this creatine phosphate system.